Hi everyone, this is the last lecture on Chapter 7, dealing with microbial metabolism. Today's mini lecture is going to cover catabolism and anabolism, which is the either breakdown of nutrients or the building up of macromolecules. This first figure is an overview of the three main catabolic pathways, and hopefully this is review for most of you. The first is aerobic respiration. When you see aerobic, you should think oxygen. That's where that last electron acceptor is in this pathway. The second one we're going to look at is anaerobic respiration. On this one, you want to think not oxygen, so without oxygen. And then the last one is fermentation, which we'll talk about a little bit at the end of the lecture. All three of these processes begin with the process of glycolysis. You can see that here in kind of a peach color. Glycolysis is the breakdown of glucose into pyruvic acid, which yields 2 ATPs. So across the board, all of these pathways will yield 2 ATPs. It's after this point that we see a change in how many ATPs are generated. If we look at aerobic respiration, that pyruvic acid is going to enter into the Krebs cycle, where we're going to generate 2 GTPs, which are similar to ATPs. And from that, this is going to go into the electron transport system. And using oxygen, free oxygen, as the final electron acceptor will create ATP. And in this one, it yields in between 36 to 38 ATPs. So this tends to be the one that yields the most amount of ATP. If we look at anaerobic, the pyruvic acid will enter the Krebs cycle and it will go into the electron transport system. There is a difference here in that it uses a non-oxygen compound as the electron acceptor. In this case, it may be nitrate, it may be other acceptors here, which yield ATP, and in this case it can yield from 2 to 36 ATPs depending on what the final electron acceptor is. And once again, this is not in the presence of oxygen. And then the last one is fermentation. Fermentation uses organic compounds, so those compounds that can contain carbon, as an electron acceptor. It ends up producing alcohols and acids, and it yields only 2 ATPs, so it does not yield as much energy as our first two processes. As we go forward, where does the cell get the materials and energy from? And in bacteria, it can be very, very different between different bacteria, but in most cases, the nutrient is going to be glucose. And glucose is broken down, as I said, into pyruvic acid by the process of glycolysis. When we look at aerobic respiration, which was the first in that figure I just showed you, there are a series of reactions that are going to convert glucose to CO2 and allow the cell to recover a lot of energy from that. It will use glycolysis, the Krebs cycle, and the electron transport chain, and in the end, it relies on free oxygen to accept that final electron and hydrogen, and that is basically aerobic respiration. When we look at anaerobic respiration, so without oxygen, this form of respiration is used by strict anaerobic organisms and also those that are capable or are facultative anaerobes. So they can metabolize with or without oxygen. It also involves glycolysis, the Krebs cycle, and the electron transport chain. This one differs, though, in that it does not use free oxygen as its final compound to accept the electrons. It uses other compounds. And then the last in that pathway, 3 pathways, is fermentation. And fermentation is the incomplete oxidation of glucose. Oxygen is not required for this, and organic compounds are final electron acceptors. So let's talk about the Krebs cycle. After we've gone through glycolysis and we've broken glucose down to pyruvic acid still has a lot of energy in it. It has bonds that can be broken and can provide energy. So the Krebs cycle takes place in the cytoplasm of bacteria, and then in the mitochondrial matrix of eukaryotes. So remember, bacteria don't have mitochondria, so this will actually take place in their cytoplasm. The Krebs cycle is a cyclical metabolic pathway, and it begins with acetyl-CoA, and that joins up with oxaloacetic acid. And then it goes through a number of different transformations. You won't need to know all of these. But the energy then is transferred from acetyl-CoA to NAD plus and FAD, and this reduces them. Remember, that means that it transfers hydrogen ions to them. Then NADH and FADH2 will carry these electrons to the electron transport chain, which is the next step in aerobic or anaerobic respiration. Through the Krebs cycle, there are 2 ATPs which are produced for each molecule of glucose, and that is through phosphorylation or the adding of phosphate to an ADP. So from the Krebs cycle, we're going to go and look then at the electron transport, and remember, this is where we have those reduced carriers. We have NADH and FADH2 that were generated by glycolysis and the Krebs cycle. These are going to pass then these electrons in a sequential and orderly fashion from one to the next in this carriers that are found in the electron transport chain. This is highly energetic, and it allows for this transport of hydrogen ions to the outside of the membrane. And in the final step of this process, in respiratory chain, so in the aerobic, oxygen will accept the electrons and hydrogen, forming water. So there are some things that you should know within the electron transport chain. There is an enzyme called NADH dehydrogenase, which should tell you that it takes a hydrogen off of NADH. There are flavoproteins, which play a role in the electron transport chain. There is an enzyme, well, a coenzyme called coenzyme Q, ubiquinone, and then there are cytochromes. And cytochromes contain these metal ions at their center, and they are actively involved in accepting those electrons and donating them to the next carrier. So they're basically a shuttle for electrons within the electron transport system. This is a really nice diagram of what this looks like. So we have our cytochromes. Remember, they can transport the hydrogens. And then we have NADH bringing that in, giving up the hydrogen, which is then shuttled to the outside of the membrane. And this is repeated over and over again along the electron transport chain. Now what ends up happening is on the outside then of this membrane, we have a concentration gradient, or excuse me, an ionic concentration gradient. So we have very positive on the outside of the membrane and tends to be more negative on the inside of this membrane. And what will happen is at this, what is called the ATP synthase, it will actually allow these hydrogens to flow down their concentration gradient, and that movement will provide the energy to transform ADP into ADP. Those hydrogens then will match up with oxygen here and form water when we're looking at aerobic respiration, and they will hook up then here with either nitrate or sulfate as anaerobic respirators. So that would be where the hydrogen goes here or the electrons go here. And once again, that aerobic respiration will yield a maximum of 3 ATPs for each NADH and 2 ATPs for each FADH. So anaerobic respiration, though, yields less per each of those. And I think we showed that at the very start, that you're not going to get quite as much without the presence of oxygen. So where does this take place? We talked about that in prokaryotes, it takes place in the cytoplasm, and that the electron transport carriers are in the cell membrane. And in eukaryotes, it's really actually in the mitochondrial inner membrane. Okay? So it's not on the cell membrane in eukaryotes, it's in the mitochondrial membrane. So with the electron chain, we see that there's that released energy from the electron carriers, and it's channeled through that ATP synthase to make ATP. And we have what is called oxidative phosphorylation, and that's that coupling of ATP synthesis to the electron transport. So I went over the fact that NADH will give rise to 3 ATPs, and FDH2 will give rise to only 2 ATPs with aerobic respiration. So what's the terminal step then at the end of this? In aerobic respiration, remember we talked about the fact that it will be oxygen, free oxygen, that will receive the electrons at the end of aerobic respiration or at the end of the electron transport. And when it ends up binding with that oxygen, you will get water. So that's the end product of aerobic respiration. When we look at the terminal step, if it's anaerobic respiration, what we see is that it utilizes oxygen-containing ions, not free oxygen, as the final electron acceptor. So in this case, it would be nitrate that would combine and accept and become nitrite, and you would have water and then NAD+. Okay, and so that's the terminal step for anaerobic respiration. And the last pathway I want to talk about is fermentation. So this is actually after pyruvic acid. There is no Krebs cycle when we look at fermentation. Once again, fermentation is the incomplete oxidation of glucose or other carbohydrates in the absence of oxygen. So it is also oxygen-free. And it uses organic compounds, those containing carbon as the terminal electron acceptor. And remember, it only yielded 2 ATPs for each glucose molecule, so it does not provide a lot of ATP. And it's used by organisms that don't have that electron transport chain. So this is their adaptation to getting energy when they don't have an electron transport chain. And sometimes when oxygen is lacking in an environment, some organisms actually have the ability to repress the production of the electron transport chain proteins so that they will revert to fermentation. And this is a way to continue making energy when there is not a lot of oxygen present. So these 3 steps, aerobic respiration, anaerobic respiration, and fermentation, are all catabolic pathways, ways that glucose or another carbohydrate could be broken down for energy. But we also want to talk about anabolism, which is the building up then of these macromolecules. And this is a very nice diagram talking about the anabolic pathways, which can happen anywhere really along the way in these paths of anaerobic and aerobic respiration. You can have the breakdown of glucose just at glycolysis, and some of that will be made into carbohydrates. Pyruvic acid can actually be used to build up amino acids, which then can also be built into nucleotides. And pyruvic acid, when it is broken down into acetyl coenzyme A, that can actually be used through beta oxidation to become fatty acids. So you can see that all along these steps of breaking down glucose, we're actually making the smaller molecules which can be put together to build our macromolecules. And when we look up here, our nucleotides will be then built into nucleic acids, which can be formed into chromosomes. Our amino acids, when put together, can be made into proteins. These proteins will work as enzymes or maybe become part of our membranes. Carbohydrates can be made into starch or cellulose as they're built up to bigger macromolecules, which can be part of the cell wall or perhaps storage for energy in the future. And then our fatty acids, when bound together, can be lipids and fats, which can make up the membrane and also energy storage. So you can see that these 2 processes of catabolism and anabolism keep that cell in a great way of either breaking down to get energy or building up to make the things that are needed by the cell. I hope that this is all pretty clear to you. If not, go back and look through this chapter. Thanks.